Hello, everybody. My name is Yesenia Villicaña Molina, and today I would like to talk about a project that I have been working on with Nelly Selim and Jaime Lopez. This is titled Topological Data Analysis Applied to SARS-CoV-2 Recombinants. Then, SARS-CoV-2 is the virus responsible for COVID-19. The genome of SARS-CoV-2 have 14 open-written frames, which encode 29 proteins. And it is made up of almost 30,000 nucleotides. So each piece of the data we have is a sequence of nearly 30,000 letters, and each letter can be T, G, A, or C. There are vertical and horizontal transfer. A phylogenetic tree is a diagram that can be represent the vertical transfer between different genomes, and a reticulate is a diagram that can be represent horizontal transfer too. But what about horizontal transfer? One of the ways horizontal transfer appears in SARS-CoV-2 is through recombination, and this occurs when two different viruses infect the same host cell and their genetic materials mixes, creating a new viral genome that contains fragments from both parents' viruses. And other possible factors are genetic diversity, RNA replication, and natural selection. But then the objective is the following: recognizing gene recombination in SARS-CoV-2 using mathematical tools from topological data analysis. But what is topological data analysis? This is a technique that analyzes the shape and structure of the data. It identifies patterns such as clusters and lobes. And the last one is very important because if you remember, when there are loops or holes, appears the recombinations, that this is the important for us. Okay, then the steps to follow are the following. First, we need an space, a way to measure distances and endpoints in the space. For example, I have 5 points in the Euclidean plane with the Euclidean metric. And for each radius, we draw a ball of radius r around each point. If two balls intersect, we draw a line segment between 2 points. And if 3 balls intersect paraways, we draw a triangle between them, like this one or like this one. And if 4 balls intersect pairways, we draw a tetrahedron between them. For example, in this figure, we have two tetrahedra. But this is something important for us. A hole appear when the radius is 1.5, and this cycle continue and disappear when the radius is 1.9, and it disappeared because it was filled. Then with this technique, we can detect the holes that appear in the data, and they represent places with potential recombination. Okay, the persistence barcode is a graph that encoding all possibles are. Then the red bars represent connected components, the blue bars represent holes, and the length of each bar is the duration time where t is equals to 2r. By drawing a vertical line at any given time, the number of red bars it intersects corresponds to the number of connected components, and the number of green bars it intersects corresponds to the number of holes at the moment. Then the applications, the methodology was the following. First, we download SARS-CoV-2 genome sequencing from GSAID. We download some of variant 1, some of variant 2, and some of recombinants of both variants. After that, we align genomes in Nextclade with the reference genome Wuhan, and we apply TDA, to align sequences with the library Goody in Python. And we conducted the experiments in two different ways. One using the complete genome, and the other one by dividing all the genome into the 14 genes that I presented at the beginning, sorry. So I'm going to present an example of evidence of recombination in one of the sets. We consider the Delta and Omicron variants. Specifically, we downloaded 23 genomes from each, and we consider 8 genomes of their first recombinant, which was named Deltacron. Then applying TDA to the complete genome was not successful, as no evidence of recombination appeared. However, we believe this is due to the imbalance in the number of nucleotides contained in each gene. For this reason, we decided to apply TDA to each of the 14 genes separately. Indeed, when applying TDA to the genes, we recognize loops in two genes. One of these in spike, the protein spike, and the other one is the ORF6. Then the conjecture after many tests is that the S spike and ORF6 genes are the most prone to recombination events, but there is still significant work to be done. Additional tests are necessary to refine the accuracy of this conjecture. Future tests will incorporate different data filters, such as by country or date, alongside through data cleaning. Thank you for your time.